Hello, and welcome to the overview of Chapter 10, Part 1. Humans define their life cycle in stages. We're born, we have an infancy, toddler, adolescence, preteen, teen, adult. Then we go into our senior citizen or geriatric phases. And cells also have a cell cycle, so you can consider it the life cycle of the cell. And that's what Chapter 10 is about. Let's start with prokaryotic cells. Because of their simple structure, their life cycle is also fairly simple. It just consists of carrying on their normal everyday activities until it's time to replicate, in which case they simply divide and go about their merry way. It happens very quickly because it's a very simple process, which is one reason why bacteria are so successful. When it's time for a bacteria to make a copy of itself, it can do so quite easily. It does this using a process called binary fission. I'll write that here. Binary fission. Bacteria, if you recall from our cell chapter, have a single circular molecule of DNA, and it's replicated, which results in two exact copies. And you can see that illustrated here. There's a green highlighted area where the replication begins, and the red area is where the replication ends. And you can read about the details, but I'm not going to worry about those for this course. Once the two copies separate, the cell begins to elongate, and the copies move further and further apart. A structure in the center, known as the septum, forms to divide the cell into two cells. It does this using proteins known as FitZ proteins. They form a ring that contracts, pinching the cell in two. And that's it. Very simple, very straightforward, very easy to do, done very quickly, and hence the reason why bacteria are so successful. The eukaryotic cell cycle is a bit more complex, but before we can go through the cell cycle and the different stages, we first need to introduce some terms and talk about the eukaryotic chromosomes. Eukaryotic chromosomes are linear, and if you recall from the nucleic acids discussion in a previous chapter, we introduced the term chromatin. I'm going to write it here for you. My stylus doesn't seem to want to work very well. Okay, chromatin. Chromatin is a complex of DNA and proteins. And the chromosomes are composed of this, and they are linear instead of circular as in the prokaryotics. Even though we previously talked about chromatin being spread out, or the form of the chromosomes where they're being used, they're unzipped in sections and being read to make proteins, so they're kind of all spread out and look grainy in the nucleus, they are still condensed into a 30 nanometer fiber in order to fit in the nucleus. So they're down to a 30 nanometer fiber. I'm sorry about the poor handwriting. And each species has a different number of these linear chromosomes. Of course, we are familiar with humans having 46 of them. And each chromosome have hundreds of thousands of genes. A gene is the code for a protein, and you know that from the previous chapters. The number has, as far as we know, no significance as far as the intelligence or complexity of the organisms. Dogs, for example, have, I'm sorry about the bad handwriting, dogs, for example, have 78. And there are even some species of rose bushes, that have 1,200 or more chromosomes. Okay, chromosomes can be categorized into two types. There are autosomes This is the first 22 pairs. By the way, these 46 chromosomes pair up to make 23 pairs. So the first 22 are the autosomes, and they have the same number and kind in both males and females. The last pair are the sex chromosomes. And they contain the genes that are responsible for determining sex. They also contain some other ones which we refer to as sex-linked traits. We'll get into those later on. The reason why the chromosomes pair up is that you get one set from one parent and one set from the other. So those 46 chromosomes end up as 23 pairs. 23 came from mom, 23 came from dad. When your cells have both sets, we refer to the cell as diploid. Di meaning two. Ploidy having to do with the number of chromosomes. Now, those pairs are referred to as homologous pairs. So let's go over what a homologous pair is. Okay, homologous chromosomes are a pair of chromosomes having the same genes in the same order. One of the chromosomes in the pair came from one parent and the other one came from the other parent. So one came from mom, one came from dad. Even though they have the same genes, those genes may not be identical. For example, on this picture of this first pair here, maternal means from the mother, so this first chromosome came from mom, and this is the paternal chromosome. The gene located in this position right here may be the gene for eye color, and on the maternal chromosome the code may be for blue eyes, while on the paternal chromosome, the one that came from dad, may be the code for brown eyes. So the genes are the same, they're both the gene for eye color, but the form of the gene may be different. We refer to that form as the allele, and we'll actually bring that term up and define it in a little bit. If a cell has only one set of chromosomes, we refer to that as haploid. This situation exists in the gametes. Gametes are the sperm and egg cells that contain the haploid number of chromosomes, only 23 pair, so that when a haploid egg fuses with a haploid sperm, you are back to the original 46 chromosomes that it takes to build a human. Scientists have been looking at their chromosomes for a long time. We've actually been able to take pictures of them when they're in a certain stage, and we can pair them up, find their homologous mate, and line them up. We've numbered them based on size, and we can determine certain things from that. And so it looks like this, the image that we take of the chromosomes. And this is called a karyotype with a K. If I can get my stylus working again. Karyotype. That's an O. Karyotype. Karyotype is actually a photograph of someone's chromosomes. They are arranged in homologous pairs from longest to shortest, and those are, you can see the numbers indicated there. And then the last pair that doesn't have a number, which is this pair right here, those are the sex chromosomes. The 23rd pair, if you will, is the sex chromosomes, and they don't look the same in this karyotype because this is a male. This person has one X chromosome and one Y. If it were a female, then it would have two X chromosomes and they would be the same size. Okay, so now we're ready to talk about the eukaryotic cell cycle. Here is a diagram of the cycle. Now, it's broken down into several phases. There's really one large phase referred to as interphase. It was originally named that because it was thought it was an intermission, and because under the microscope it didn't look like anything was going on, but it turns out the cell's quite busy during this time period, and so we've since then divided it into three phases known as G1, S, and G2. Then we have the M phase, which is referred to as mitosis. It's divided into 4 or 5 phases, depending on which textbook you're looking at. And then we have the C phase, which stands for cytokinesis. The cell cycle varies greatly in length among cell types. Some cells will not even go through all of these phases. It depends on the type of cell and its function. But we're going to cover all the phases so you know all the possible phases that a cell could go through, but just bear in mind that the length and what phases a cell actually goes through may vary. So G1 stands for the primary growth phase, and this is where when a new cell is first formed, it begins G1. The cell grows. It starts out life as about half its mature size, so it grows until it reaches maturity. It carries out its normal functions, and this is considered the longest stage of the cell cycle, even though it's not illustrated as such here. Now, some cells never leave this stage, so if they don't ever leave this stage, then instead of calling it G1, we refer to it as G0. The S phase stands for synthesis, and this is where DNA is replicated. And we will go over that shortly. G2 is the second growth phase, where the cell is getting ready to divide. It's gotten a signal that it needs to divide, and it got that before it went into the S phase. That's what told it needed to make a copy of the DNA. So now it's adding materials and making proteins and structures that are necessary for going through this cell division. Then mitosis is the stages of replication of the nucleus, and cytokinesis is the actual division of the cell into 2 new cells. So let's look at the details. There isn't really much. We've already gone over G1 and G0, so let's start with S, the synthesis of DNA. So the cell gets a signal that it needs to divide, so it'll go into S stage. Whoops, sorry about that. And replicate its DNA. And we talked a little bit about this when we introduced DNA as a nucleic acid. So we're just going to review here. DNA is replicated in a semi-conservative method. That means the original strand is unzipped, and each original side of the ladder, if you will, is copied so that the 2 new strands are half old and half new. So the new helices, if you will, contain 1 old strand and 1 new strand. Replication begins at RNA primers. If you look in this image to the right, here you have the RNA primer, which is this little yellow area sectioned off here. So it's unzipped, and then these RNA primers are the site of where replication begins. Now, there's a number of enzymes and proteins involved in this process, but a major category of them are called DNA polymerase. So I'll put this here, DNA polymerase. Okay, this should be an R. So DNA polymerase enzymes match base pairs to the existing strand and links the nucleotides together. And that's the blue segment you see that's being built in the image. So this area here. The bases are always added from the 5' to the 3' end of the strand. So one strand, which is referred to as the leading strand, is built continuously as it unzips in this direction. But the lagging strand has to be built discontinuously. One section will be built because it's going in this direction, and then it sort of has to wait for the DNA strand to unzip a little bit more till it can do the next section. Okay, so we're going to end this video here, but we're all set for part 2, where we're going to talk about the replication of the nucleus and then actually dividing the cell into 2. Thanks for watching.